From Microbe TV, this is Office Hours for Wednesday, October 26, 2025. I'm Vincent Racaniello, and welcome to your corner of the internet where you can learn about viruses and also some science in general, why science is so important to the world, including the U.S., and we'll give you the straight facts. Let's start off by thanking our mods tonight, doing double duty, some of them. They were on TWevo this afternoon. Les is here. Welcome, Les. Barb Mack is here. Steph SF is here. And Tom Steinberg is here. Thank you for joining us and keeping this a civilized corner of the internet. Welcome, everyone else. Tell us where you're from. We will shout you out before we get into the agenda. What am I going to do today? I want to talk about some things in the news regarding science and science policy in the U.S., measles in Texas. Oh, my gosh. And then we'll have a mini lecture. All right, Dagfinn is back from Norway, where it's 3 a.m., 4 degrees Celsius. Boy, you're brave to stay up. I'd be asleep at 3 a.m. So thank you for joining us. It's appreciated. Jeff is in Elgin, Illinois, where it's 49 and very nice. Artemis is in Montreal, 35 SF. Wayne is in Urbana, 54 F12C. Gallbladder 8 is in Baton Rouge, where it's 26C. Kip and Laura from San Francisco, thank you for your support of science communication. Sorry I missed you in New York. I did see you one day, but the other day is just too packed. Sorry. Next trip, Kang is in Chicago, where it's 5 degrees and clear, a little chilly there. Elizabeth is in 8C, West Virginia. Visto is in Sydney, 23C and sunny. Carol, our favorite nephrologist, in sunny Bakersfield, California, 28C. So that's the centigrade where you could just flip it around and you know the Fahrenheit. 28 is 82. Hello, Brian in Montreal, 1C and almost clear. EHS Talent is in Lehigh Valley, PA. Cheryl is in Santa Rosa, California, where it's cloudy and 57F. Rainey Parker is in eastern North Carolina, where it's 58F. Here in New Jersey, by the way, it's chilly. I don't have my phone to tell you the temperature, but I still am wearing a heavy winter coat, a scarf, and a knit cap and gloves. It's too chilly for me. Sorry. Maureen is in Northeast Ohio. And Patricia's in D.C., where it's not so nice today. I'm sorry about that. Eliza is in Ontario, 3C. Hello, Tracy from Chapel Hill, where it is 12C. Truth or Dare is from La Jolla, California, of course. Pete is in London, where it's 10 degree C. Fixing a leak. John says it's 10C in Minneapolis tonight. Silent Rudder is in Wichita, Kansas. Nice 23C today, but hazy and smoky from fires in Oklahoma. Richard Schoenbaum, El Segundo. Nice to see you, Richard. Outside of D.C. is MN, and it's chilly. Terry is in Two Rivers, Alaska, where it's 4 degrees Celsius. Mitzverdi has put his flag there. I don't know what it is. I'm sorry. Well, we could look it up. blue, white, red flag. And it's vertical. France! That's the national flag of France, surrounded by viruses. Welcome. Meet. Betty's in Washington Heights. Nice to see you again. Lugotic is 3 degrees C in Chicago. Rach is on the Isle of Man, where spring has sprung, 13 C and sunny. Kang says, unfortunately, lies have better marketing than truths. Could a mini lecture on the germ theory of disease counter the miasma of misinformation? We could do that. We could do that. I was thinking of getting Carl Zimmer on Twiv to talk about his latest book where he talks about that also. Hat is in LA, where it's 60F. Abdul Aziz is in KSA, 13C. Mike is in Seattle, 61F. Thunderstorms, maybe tomato. Sorry, tornado. Carmen is in Costa Rica. Carmen of the Sea. Well, that would be in Italian. I don't know if it's the same. Tractor Poodle is in Bangkok, where it is 20. 7C. Very hot, yeah. Mitz says it's 9 degrees C in the Alps. Markle is on the east end of Long Island. 40F. Elevated risk for wildfires. Elicio is in Belém, Brazil. 30C and rainy. Very hot and humid. Julie S. is in north central Pennsylvania. This is what we're going to talk about a little bit tonight. Zan says, I keep hearing some people tout vitamin A, but no one has mentioned you can overdose, leading to liver toxicity. Well, that's just what happened in Texas, and we'll talk about that. It's gross. It's just gross. This guy should be sued for medical malpractice. Katya is in Detroit, Michigan. Welcome. SRR is in Paso Robles, where it's 61F. Abdul Rahman is in Manhattan, where it's 9C. I left Manhattan not too long ago. Chris is in Portland waiting for severe thunderstorms. And Heather is in Western Canada. Pete says, I remember Dan Wilson take on a virus denier. It's just not worth it because they would just lie. And it's just not worth countering someone who lies all the time. There's nothing you can do. No matter what you say, they will say you are conflicted and they will lie. So not doing it, folks. Sorry. Patricia in Massachusetts, welcome to office hours. Jean-Claude, 8 degrees C in Bern, Switzerland. I've been to Bern once. Check the level on mics. What is it, too high? Is it too low? Is it just so? What's the Goldilocks on the mics? If anyone else has a problem, let me know. Anyone else here? Yeah. Arguing with these anti-science people is like arguing with a toddler. It's just, you're not going to get anywhere and it's not worth it. And you end up looking foolish. So I know we're right about the science. Helen is in Western Massachusetts. Hello, Helen. Mike sounds good. If it's too high, lower the volume. Thanks, everyone, for responding. I appreciate that. All right, so let's see what we can do here with a couple of news articles. I've queued up a couple. Ooh, here is Questella in Ohio. She's here. Welcome. Silvio Pina. Bolognese in Burbank. That's great. Italian flag and the American flag. At least I don't have to look those up, right? Let's see what I can do here with these links. All right, what should we talk about first? Yeah. Copy link URL. All right, so I'm going to paste this link. Unfortunately, it's a New York Times, but I will bring up the article myself and we can discuss it. Okay, Chrome. And we'll put the URL in. All right. Oh, come on. I've already logged in multiple times. Sorry, folks. I got to find my password. New York Times. That's not it. The wrong account. Let's see if we can go to Safari and get it properly. Otherwise, we're going to have a problem. Such a pain. Nope. Nope. My damn password isn't showing up. Oh my gosh. This is tragic. Well, let me guess. Nope. Oh well. Can't get into my Times account, which is ridiculous because I've been logging in. Yeah, there it is. Account. I'm in. Well, I'll just find the article here and we'll discuss it. Sorry about that. This is how you accidentally invite the Atlantic to the group chat. Yeah. All right. So this is a New York Times article, which came out today, I believe. Remedy supported by Kennedy leaves some measles patients more ill. So you remember he promoted vitamin A as a measles preventative and cure. And so what do you think people started doing? They started taking vitamin A or giving it to their kids. And they gave them too much. Vitamin A is a fat-soluble vitamin, concentrates in organs, and it's known to cause liver disease. So doctors in West Texas, this is by Teddy Rosenbluth. Doctors in West Texas are seeing measles patients whose illnesses have been complicated by an alternative therapy endorsed by Kennedy. So these parents are turning to supplements, right? They think that they don't want to get vaccinated because the anti-vaxxers say measles vaccine is no good. So they took vitamin A. They also got cod liver oil. And they have signs of liver damage, some of these kids. Okay? So I knew this was going to happen. I knew it. So Kennedy said on a Fox interview, he's heard of almost miraculous and instantaneous recovery with vitamin A. That's wrong, okay? First of all, vitamin A is useful to prevent severe measles in resource-poor countries where kids are starving. It's never been shown. It's never been shown to do anything about measles in the U.S. And it's not a preventative. It's not going to prevent infection. You're still going to get infected. So the idea of recommending measles is bogus. If he were a doctor, he could be sued for medical malpractice. This is disgusting. And he should be out there pushing vaccines and saying, this is safe. All these things I've been saying all these years are wrong. You should get these vaccines. But no, he doesn't want to give away millions of dollars of income. So we know where his priorities lie. But buried in this article, down towards the end, is a very interesting passage. So Kevin Griffiths wrote an opinion piece for the Post, the Washington Post, on Tuesday. He used to be the communications director for CDC. He resigned because of how Kennedy is handling the Texas measles outbreak. He writes, quote, "In my final weeks at CDC, I watched as career infectious disease experts were tasked with spending precious hours searching medical literature in vain for data to support Kennedy's preferred treatments." I find this disgusting. It is not a miracle treatment. There's no antiviral for measles, and it causes liver toxicity, as you can see. So that's our first story here. Let's see what people are saying. Okay. Just a few more people have arrived. Spamlet from Bedfordshire. Welcome. Laurie is in Charlottesville, 9C. Angela O'Kelly is in Ireland. Edward is in Slovakia. Bridget is here. Welcome. Okay, here, Bad Cat is in Winthrop, Maine. Okay, now there were two other stories I wanted to talk about, but Dagfin posted a link to one that I wanted to talk about also. Where is it? Oh, not a link, but another thing. So, RFK wants to reinvestigate the link between MMR and autism, MMR vaccine, which has been investigated over a dozen times. No study has ever found a link. So he's going to waste money. But you know why he's doing this? He's gotten David Geyer, an anti-vaxxer on his own and not a scientist, to lead this study. So it's clearly going to be a bogus study. And he will somehow come down saying, yeah, MMR causes autism. We need to study this more. And it will cause less and less, fewer and fewer people to use the vaccine. And we are headed for a real problem in this country. It's a real problem. So Dagvin says, how is this study going to impact American science reputation? Well, it depends on how it's presented, right? If it's presented as this is Geyer leading this study, most scientists are going to just trash it. And David Novella at Science Based Medicine has already started to do that. He said, I won't believe any study led by David Geyer. He doesn't know what he's doing. So RFK Jr. taps this guy to do a meaningless study. So he's going to waste money. Yeah, so some of that money, you're cutting from NIH, he's probably going to use to do this. It's just disgusting, folks. Just disgusting. There was another article here. Unfortunately, I can't. Let's see if I can get the URL. Yeah, and that's the problem is, sorry. The problem is that people will believe it. Many people will say, I knew all along that there's a problem here. I want to see how they're going to do this study. It's going to be so crappily designed, you won't believe it. Okay, let's see. Les put in another link here. Oh, yes, here's another doozy. U.S. to end vaccine funds for poor countries. This one is unlocked, so he put put a link in the chat, and it's unlocked if you want to go look at it. So here we go. I can show you this on my screen share. U.S. to end vaccine funds for poor countries. A 281-page spreadsheet obtained by the Times lists the Trump administration's plans for thousands of foreign aid programs. This is by Stephanie Nolan. So they're going to terminate the U.S. financial support for Gavi that's helped to purchase vaccines for developing countries for 25 years. Gavi is it's a vaccine alliance. I don't actually know what it stands for. Global Vaccine Alliance, something like that. Global Alliance for Vaccines and Immunization. Jeez. And they have, been giving vaccines to kids for years, 25 years, and they're going to cut back. They're going to eliminate those programs, terminate, and scale back malaria as well. They're going to continue some grants for HIV and tuberculosis and some food aid, okay? But I don't understand this logic, right? I don't understand why you need to be mean to people. For years, the U.S. was able to help people in poorer countries, and we should do that. Why all of a sudden do we not want to do that? Because one person and one administration thinks it's a waste of money. Do you think most Americans don't want to help the rest of the world? I don't think so. This was never mentioned in part of the campaigning, right? So I just think this is duplicitous. So anyway, this is... this is congressionally allocated. It's not clear that they can end the programs. And I'm sure it's going to be litigated in multiple courts. And this is going to drag on a long time. Meanwhile, kids will die. And no one in this administration cares because they all care about themselves. Okay. It's very frustrating, folks. I hope you're as frustrated as I am. Let's see what people have to say about this. All right, so Jeff says, vitamin A is effective, to prevent mortality in under-resourced countries. It's never been tested in the U.S. Not clear it would do anything. We're not under-resourced. Oy. Bridget says, I appreciate the astonishment from others in the last TWIV about the remark that NIH has done nothing, which is simply absurd. Yes. Instantaneous recovery by vitamin A administered by Santa Claus or the Tooth Fairy? What's going on? Indeed. The U.S. is now a laughingstock under Republican authoritarianism, and the GOP has shown that the U.S. can't be trusted. There are a laughingstock. countries are laughing at them. Well, there's the security signal screw up. So amateur. And, China and Russia are probably licking their chops. And, it's not safe to be here anymore, in my opinion. We can't trust the GOP at all. They lie all the time. Tom says vitamin A is certainly not as antiviral in appropriate doses. It supports general health. Jonathan is an Encinitas. You missed talking about vitamin A causing liver disease in children, and then the administration ending vaccines for poor countries. Okay, here's another one. Oh, yeah, this is wonderful. Maybe Les can put in one of those free linky thingies. I'll paste it in here. So this one is also a science article. Maybe I don't need less to look at that one. Maybe it's unlocked. Anyway, you got the link there, whatever you want to do with it. Saying the pandemic is over. So let's clear up this screen here and share this. Saying the pandemic is over, NIH starts cutting COVID-19 research. That's it, new target. 29 awards made by NIAID, including 9 part of an antiviral drug program to prevent future pandemics, all canceled. The end of the pandemic provides cause to terminate COVID-related grant funds, the notification states. These funds were issued for a limited purpose to ameliorate the effects of the pandemic. Now that the pandemic is over, the funds are no longer necessary. That's just an outright lie. Okay, they were not issued for a limited purpose. They were issued to do research to try and prevent a future pandemic. Now that the pandemic is over, the funds are no longer necessary is bullshit. Are we going to terminate all flu grants because the 2009 pandemic is over and that's 1968 and 1957? Stupid excuse, simply ignorant. And this antiviral drug discovery center includes Charles Rice, 9 U.S.-based effort to develop new drugs, and also against not just COVID, but Rift Valley fever, filoviruses, flavies, paramyxo, picornas, togas. And as Charlie Rice says, Nobel Prize winner at Rockefeller, and he echoes what I just said, the termination has a misleading rationale and a pointless, ill-advised move that will hurt U.S. science and pandemic readiness. So there you go. There's another example of useless, ill-thought-out cuts of science. Science projects are funded so that you can pursue some innovation and you can try things, you take a risk or something. And that's what these were. And they weren't aimed at taking care of the COVID pandemic. They're aimed at the future. But that's an excuse that these people are writing when they say that. It's an excuse. So what's going to happen to this money? I don't know. But I'll tell you, they say, the White House press secretary says, this is in line with the president's desire to focus on chronic diseases. Right? So this is not a chronic disease. This is BS in capital letters. There is no intention to focus on chronic diseases. It's a lie. They're going to just give the money to someone else, and you know who that might be. If they were going to focus on chronic diseases, the way the NIH works is they decide to investigate a new area. They put out a call for research proposals. And they have a long, detailed description of what they want investigated. And then you write a grant proposal that addresses these. We will never see such a call for proposals on chronic diseases. Never. Well, if they do, then I'll eat crow. I'll say, yeah, I'm wrong. But they're lying about reappropriating this money. And I find this disgusting. I can't find a strong enough word, but it's so gross. I just hate when people transparently lie about things. All right, so we had the vitamin A, we had the cutting of COVID-19 research, and we have Gavi, Cuts and Gavi, the Vaccine Alliance. So Bridget's in Evansville, Indiana, where it's 13C and Pleasant, Angry Penguin. Yeah, press the like button, folks. Let's see who we have here. We have 200 people and 100 likes. Press the like button, folks, please. Thank you very much. Silvio Pina missed a lot of TWIVs. I wonder if Amy's federal job is safe. I can't comment, Silvio Pina. I'm sorry, it's not. I can't do it. Ariozone is from Asheville, North Carolina. Yes, it's a nightmare. The whole thing is a nightmare. Rip de Comp, welcome. Good to see you. Julie says the use of vitamin A to prevent or cure measles is not evidence-based. There's no evidence in the U.S., but in countries where kids are unhealthy, it can reduce the severity of measles. That's well known. Yeah. The new nomination for head of CDC, Susan Monarez. Got to look into it. I don't know her. And I will look into it and report on it. Probably Friday's TWIV. And if anyone knows anything, please share it. That would be great. Cassie is in Southern California, 64F. Yes. If at the FDA can't purchase reagents to do the research, same case with NIH, I believe, right? Yeah, the purchase cards have a limit of $1, and they didn't realize that these are used to buy supplies. That's how you purchase lab stuff these days. You don't do purchase orders anymore. So they just cut it off, and nobody can do research. Yeah, you run out of your stuff, and that's the end of it. Lollipop Doc suggested getting pneumococcal vaccine. Yes, Daniel Griffin always says get it. And, there are a couple available, and the one with the most different antigens is the one that you want to get. It basically protects you against getting severe disease like a viral vaccine would. Tractor Poodle says, if RFK Jr. includes the right people in a Signal chat group, the MMR study should go very well. Yeah, include the editor-in-chief of The Atlantic. I think it's a good idea. There's no logic. I think they just want to redirect all the money to wealthy individuals in the U.S. Like I said, I don't think they're going to redirect it to chronic diseases. Not happening. It's going to go to wealthy people. They're going to get a tax cut, and then that's where the money's going to come from. So this is so obsequious. You compromise the health of everyone in the U.S. to allow wealthy people to get wealthier. Well, what's the name of that, right? Oligarchy. I don't know. It gets worse every day, folks. I'm very frustrated. Kip says, if we were vaccinated 60 years ago for MMR, so 60 years ago, that would be 1960. That would be 40, 1960. You would need to be vaccinated twice unless you caught measles. If you were living in the 50s, then you definitely caught measles and you don't have to be vaccinated. It's actually a very good chart that Daniel published on the last clinical update, which tells you, well, why don't we bring it up, right? Let's bring it up. This is what this is for. Microbe TV. I'm so glad we have this nice website with all the podcasts on it. Come on, show up. Where is TWIV? I don't particularly like this front page. It's hard to navigate. This week in virology. Okay, Daniel Griffin. Where is this vaccine thing? I'm just going to look in my show notes. The hell with this. Can't find it. Oh my God, who makes these show notes? Be fired. TWIV archive. Clinical update. 1202. Here we go. All right. This is beautiful, actually. Let's blow it up. Let's screen share. Let's blow it up some more. Here we go. So, measles vaccination recommendations by year of birth. Okay, so now, Kip, this is for you. If you're born prior to 57, you had measles as a child, nothing to do. If you're born in 57 or later, if you had 2 doses, you're good to go. If you're vaccinated between 63 and 67, when a less effective vaccine was used, you should get 1 dose. If you're vaccinated between 68 and 89, get another dose. It was only 1 dose recommended at the time. If your vaccination history is unknown, 1 dose. Or get titers and go by that. Okay? Thank you, Daniel Griffin, for that. So you got that, Kip? Nice question. Love the comments on the live stream. Too bad we can't like the comments. Yeah, I'm sorry about that. Unfortunately, Ariozone is right. All these cruelties are written in the Heritage Foundation's Bible, Project 2025. Yeah, we saw it there, but during the campaign, Trump said, I'm not going to go by that. And then, of course, the minute he's elected, he's going by it. Another lie, and nobody cares who is voting for him. Nobody cares that he lies left and right. But they are cruelties. That's the perfect word. They are cruelties. And it's totally changing the face of the U.S. But, folks, there are people like us here tonight who care about others. We're still here. We're probably over 50% of the U.S. population. And somehow we're going to fight back. Could you share your expertise on foot and mouth disease caused by foot and mouth disease virus? Emergence of foot and mouth disease in Central Europe after 50 years of being disease-free. So, foot and mouth disease virus is highly contagious. It's known to travel across the British Channel, the English Channel, sorry. Travel across the English Channel and infect cows from farms in the UK to infect farms in Europe. In the UK, when there's an outbreak on the farm, they cull all the cows because it's so contagious. So you should consider that, but also vaccinating can be useful. There's a foot and mouth vaccine. But yeah, it's a big problem for cattle industry because you can't sell cows that have foot and mouth. Piñata, where you're frustrated enough to get 3 or 4 other people to vote with you. Only 49% of your voting eligible population even bothered to turn up. This is a big problem. It's exactly right. 51% of people didn't vote, and that could have made the difference. They're used to the niceties that they get here in the U.S. Well, I got news for you, 51%ers. They're going away, and only wealthy people are going to have them. You need health care? Nope, not happening. Only if you're really rich. Next time, get your ass out and vote. Hey. Parker says, King of Unrelated. It's okay. Does the contents of a vaccine actually affect how much it hurts or how gentle it's given? Typically, well, it varies from person to person, but what happens is when you inject the vaccine, say in your deltoid muscle, it's being recognized by the immune system, and the immune system is being fired up, and that's what causes the pain. So some vaccines have adjuvants, which are used to stimulate the immune system even more because that gives you a better immune response. So they can hurt more. And so, people say the Shingrix vaccine hurts a lot. I got 2 doses. I didn't feel anything. For my first COVID vaccine, I got a fever for a couple of hours. And then the next 2, I felt nothing. So it really depends on people. Some people everybody makes a different immune response. Yeah. But, you can have an unadjuvanted vaccine and still it provokes a nice immune response. So adjuvants will make the side effects worse, so, pain at injection site and so forth. But you can get them without adjuvants too. Haley says the administration is under-resourced in brain cells. So Nell said today, confident but not competent. They're all competent. They're all confident that they know what they're talking about. They speak so authoritatively, and a lot of Americans buy it, but they are incompetent. Yeah. Patricia says, it's true we're the laughingstock, but countries are copying us and being even more authoritarian than we are. I thought we were copying like Hungary and Russia. There's still going to be non-authoritarian countries. I hope. Rip de Camp is in Taos. First few nights without ice. Good to have you. I'm not going anywhere. I'm staying here. I think it's. I don't even want to fly anymore. I don't trust the FAA. So I'm staying here. I'm just going to do my podcast and speak up as much as I can and try and make a difference. I'm not leaving. As the world turns. I remember that. Soap opera. Yeah. My grandmother used to like to watch. Hat gives an article in the Lancet on the efficacy of RSV vaccine in Great Britain. I would be surprised if Daniel talks about it tomorrow. Elizabeth says at least people see the lies now. So you think the Signal incident is now making people see the lies? Culmination of crap started by Reagan. It's true. Before Reagan, Republicans were way more reasonable. But this bloody actor who knew nothing screwed it all up. Bad cat. Before I could get COVID vaccine, I got MMR because research showed that those receiving MMR exhibited 50% risk reduction in COVID symptoms. Yes, this is true. So any infectious vaccine probably had this effect. It's a couple of months, it's not forever. But I also said to people, consider getting MMR. We don't have a COVID vaccine yet. And it'll protect you for a couple of months. Chumakov wanted to do a clinical trial with oral poliovirus vaccine because he thought the same thing would happen, but that never happened. You think the GOP will end flu research funding? Yeah, flu research. Oh my gosh. That's a lot of labs. They're pouring a lot of money into it because the pandemic is over, right? I wonder if the strategy is to have all Americans sick, and then it's easy to take over the whole country. Well, I'm not going to be sick. Stay with me, folks. Yeah, according to RFK, we talked about this last week. He wants to let H5 run through the clocks. That's just a dumb idea. No basis. We talked about that last week. No more chicken or eggs. What if one chicken lives? You're not going to be able to do much with a chicken that's had H5N1. Nobody's going to buy it. That's the problem. And you won't have. It's 100% fatal. And if one lives, it's probably not a genetic basis. There's just something else. Yeah, Eliza, exactly. Long COVID is a chronic disease. So that's getting cut as well. Daniel mentioned this a couple of weeks ago. Columbia had a long COVID program partially funded by CDC, and CDC said, nope, no more funds. We're cut. You're cut. Kakistocracy. I haven't heard that word in a long time, right? Governed by the least suitable or competent citizens of a state. The worst, least qualified, or most unscrupulous citizens. Can you believe that we have to have a word like that in our language, even? It's gross. Bridget, yeah, we know you can't discuss Amy's situation, but we do miss her and her sweaters. She was fantastic, and you two made a great team for office hours. Yeah, I can't wait to see the Republicans tackle chronic disease. Well, I say they're not. They're not going to even bother. Doctors and nurses did not cause metabolic disease. The way to tackle it, though, is to regulate our food supply better. But do you think RFK will? No, of course not. He said the other day, eating fast food is the solution. Oh my gosh. Yeah, if I recall the latest clinical update correctly, Daniel said there were roughly 4 times more deaths in 23-24 from COVID compared to flu. COVID research is still relevant. Of course it is. What they use as an excuse is pure BS. They're lying. Of course it's relevant. They just want to take the money and do other things with it. And, they say the White House secretary says we're going to use it for chronic diseases. I don't believe a word of it. I don't believe a word of anything that these people say. The guy Hugspeth or whatever that. We didn't do anything on Signal. Lying just outright. Then he got caught. John says, I picked a bleak time. Yay. To renew my interest in life sciences, but that won't stop me from stumping for wherever I can. Thank you, John. This is what I think Areo's own. The money they're taking from necessities are intended to cover tax cuts for the oligarchs. Yep. H. Sue in Austin, Texas. Welcome. I don't want to talk politics. I try not to. But they're mixing it up with science. I suspect they want poor people to die. It's a scary thought, but I cannot convince myself otherwise. Yeah, I guess obsequious was not the right word in the context I was using it. Yeah. Yeah. They want to go back to 49. I think they're headed for 29 before FDR. Yeah. You know, it's like Hoover said, oh, the crash will be over soon. Liar. There's a great column by Helen Cox Richardson about this lady who made her way to be Secretary of Labor for FDR and did amazing things. You should check it out. Hello, Jill in Wilmington. Nice to see you. Hard to stay sane. We try. The middle class was created by the income tax with its high upper marginal rates. It ended the Gilded Age because wealth aggregates without control. It doesn't trickle down. Yet, Reagan said we're going to do the trickle down theory. Just lied. He knew it wasn't going to work. It was just a lie. Hello, Amy, wherever you are. Ray's says, I struggle to find show notes on Dr. Griffin's TWIV. No, it goes up Saturday morning at midnight. Microbe.tv/TWIV. They're all there. I mean, now and then it's later because Daniel is traveling, but the notes are always up. You couldn't find the chart. Yeah, neither could I. It should be in the video, actually, because our editor puts them in. But I don't know where exactly that came from. I think that was New York Presbyterian Hospital. Yeah, it's a good chart. Elizabeth, not to be too conspiracy theorist, but I generally believe this is a combination of rich idiots wanting to be oligarchs and end-of-days cultists wanting to hurry the end. Scary stuff. Hello, Will from Changsha, where it's 27C, and yesterday it was 36. That's pretty hot. Julie said, I was born in 56. Well, then you had measles, for sure. Most people got measles, so you got it. That's according to that chart from Daniel. Yeah. I think polio scares me more than measles. It's a toss-up. Polio can't kill you, but it also paralyzes you forever, right? We'll see polio in the U.S. I hate to make such proclamations, but we will. Areas on mumps doesn't seem to come up, or rubella. We could do a mini lecture. Yeah, let me write it down here. Mumps mini lecture. Okay, written down. See, I was born in 53, and I don't remember having measles, but I must have. And so I'm not worried about getting vaccinated. Tractor Poodle asks, is there evidence that people are not having their pets vaccinated because of RFK Jr. and anti-vaxxer influences? I don't know. It's a good question. Let's look it up. So people are generally immunizing dogs and cats with core vaccines, but there's some hesitancy with non-core vaccines, and that comes from human vaccine hesitancy. So the core vaccines are rabies, canine parvovirus, and feline panleukopenia. So there you go. So as usual, it's a mix. Nicole says, U.S. scientists need to be louder than misinformation. more social media, more interviews on popular podcasts, more news presence, etc. I'm not seeing high-profile scientists fighting, except you. I'm not even a high-profile scientist. I don't do research anymore. I agree. And you know what, Nicole? Now that they've lost their grants, they'll have some time to do this. Because they always had this excuse, I'm too busy running a lab, I can't do this. Well, now they have time to do it, and they need to. Bridget says, I went to subscribe to ProMed email update. So ProMed is a disease outbreak tracking service under a new email. And PubMed info via NIH is having some issues. I guess research isn't important. I'm a little upset by all of this. I think, as I said today on Twivo, most people in the U.S., many people do not realize the importance of basic research to their health. And we need to impact that. I mean, I try as much as I can, but I have a small reach, right? We need more people to be doing this. H2 says, if I am a negative strand virus and am alive, do I have positive strands in my living environment? Well, yes, if you're a negative strand virus, at some point, you need to make mRNA to make proteins, and mRNA is positive stranded, right? So the interesting thing about negative strand reverse genetics, so for poliovirus, you just clone the genome as a plasmid, and that's infectious. But for negative strand viruses, you had to express the plus strand RNA in cells to make all the proteins, and then that got infectivity going. But yeah, you have plus strands because you need to make proteins, mRNAs, right? Will is in the middle of reading booster shots. Good, I'm glad you like it. Glad you like it. Ted asks, does everyone who gets measles experience immunity amnesia? No, it's, the number in the study done in the Netherlands in 2019, I know this because it was in my lecture today, 11 to 70% of children had immune amnesia. So not everyone, but, up to 70%. It's a lot. So I don't know the reason for that. The reason that it occurs is that measles virus infects lymphocytes and destroys them, and that memory B and T cells are destroyed. But why there's such a variation, I don't know. And Bridget said, now we can't have nice things, and the rest of the world knows we can't be trusted. Yeah, it's unfortunate. Many people in the U.S. can be trusted, right? But the country, if you're dealing with the country, politically or economically, forget it. They're just lying left and right. But people are lying to them, too. I mean, this Trump-Putin vibe, man. I don't know who's on top there. I don't think it's the orange guy. Hello, Martha from South Carolina. Nice to see you. Tatiana says, Republicans successfully suppressed my vote by making it too hard to vote in absentia. Yeah, that is their solution. You got it. And unfortunately, the Democrats don't fight back hard enough. Really pissed off at them. Pete says, UK had a major foot and mouth outbreak in 2001. 6 million cattle and sheep slaughtered. Yeah, very sad. Kang says, unfortunately, experience is a hard teacher, and many people only learn the hard way. That's right. It's absolutely right. They're learning it now. Jeff writes the book Strangers in Their Own Land by sociologist Arlie Russell Hochschild explains those that are the teabaggers, MAGA, as those people who feel disrespected. They are horrible people. They're disrespected because they bring it upon themselves. Eliza's also enjoying booster shots. Great. I'm glad a bunch of you went out and bought it. Very good. Wow. Bridget got Shingrix, COVID, flu, and Tdap in one go. That's very good. Your arms must have been sore. Hello, Lise from Columbus. What's the strategic plan among the scientific community for fighting back with respect to Trump's cutbacks to grants, etc., to science? There is no strategic plan, and that's part of the problem. I don't want to criticize my colleagues, but they've been very complacent for many years getting NIH money. They never made any plans what to do when it was taken away from them, because it's a government subsidy basically, and it can be taken away, as we see. And now they're scared, right? But there's no plan. They have to have some kind of a plan. They need a leader to coalesce around. Someone who can say, we need to do this and we need to do that, come up with a plan, have demonstrations and so forth, but not happening. This is very funny. I've hardly felt anything from injected vaccinations, but stubbing one's toe is extremely painful. It is. It is. Yep. From Helen, avian flu question. Do we know how many mutations or which would be needed for H5 to allow human-to-human transmission? No, we do not. What's the natural mutation rate? It's the same as any other influenza virus. It's probably one mistake in 10,000 per 10,000 bases. Very, very error prone. But, okay, so 2015, two groups took H5N1 avian influenza. If you infect ferrets with that virus, it kills them, but it doesn't transmit, like from cage to cage. So they selected for a virus that could transmit through the air from cage to cage. And that virus did not kill them anymore. And it had a certain number of mutations. Many of them were in the H5 hemagglutinin. And they influenced receptor binding and also the stability of the protein at low pH. Those mutations, those amino acid changes have been seen in circulating isolates. So it's not clear what that tells us. I mean, I think there were 5 or 6 that were needed, and that's not just one. So you see, the acquisition of transmission led to a loss of pathogenicity. So I don't know how that applies to people at all, if at all. We just need to monitor the circulating viruses and see what's going on, see if they're becoming transmissible and what's their virulence. Juliet says administration technique for IM can also affect how much it hurts in volume injected. Yeah, the flu vaccine, it's like 1.5 mLs. At the end there, it's like, pain. It's just this kind of vague pain of putting a lot of fluid into your muscle. That's what it is, yeah. But I didn't find a post-vaccine pain. It was just that vaccination, yeah. Polka Dot Pup. I've watched TWIV since the beginning of COVID and looked up the papers in the show notes. I've learned so much. I'm really glad you like it. I really appreciate that. It makes us feel good that we're doing something, right? H2, thank you for your contribution to science communication. You are making a difference against the widespread nonsense. Thank you for what you do for all of us. You're welcome. It's it's what we love to do, and we'll keep doing it, especially with supporters like you. Really appreciate it. Look at this. Have you read George Washington's farewell address? I have not. He warned that if parties were allowed to capture Congress, human nature of tribalism would inevitably lead to the rise of a dictator. Huh? Well, we have a party that has captured Congress, and I do not understand. So, is it really that important for you to have a job? I mean, a senator or a congressperson doesn't really make that much. Is that really important to you to violate all sensibility and ethics in this country? I guess it is to them. But you know, when we have a dictator, there's not going to be a need for a Congress, so you're out of a job. Think about that. Yes, that having nice things comment from Bridget. That must have come from somewhere because it ranks to me too from maybe William Gibson. Yeah, yeah. And that's it. She says I've read a lot of Gibson. Yeah, I have too. I really like him. Who's going to treat our oligarchs so there will be health care for rich people? Of course. You know, exclusive hospitals and so forth. Hannah said, I'm a toxicologist. I took care of a man who accidentally overdosed on hydroxychloroquine. He was taken for COVID. He was intubated for several days. Idiot. The vitamin A stuff likewise makes me nervous. He won't listen to voices of reason because he thinks they're lying to him. He has to listen to liars. Tim Snyder says this is a kakistocracy until a future historian coins another name. Ay. Authoritarianism necessarily breeds incompetence. All right. H5N1, is it 50% or so fatal to humans? So, you know, there's just been around 1,000 cases, 890 since 2003 in 23 countries. And let's see, fatality rate, yeah, it's about 50%. It's quite right. I mean, you know, it doesn't transmit among people, so it's good. It does kill them at a high level, yeah. Les says FDA is Food and Drug, not HHS. No, but it is under the guidance of the Department of Health and Human Services. Yeah. Isn't diabetes a chronic disease? Of course it is. Yep. You can sometimes control it with medications, but it's always there. Yeah. Their idea for tackling chronic disease is to blame the sick person. They will 100% double down on that. And we need much money for that, right? I shudder to think of what the next pandemic is going to be like. Look at the measles outbreak in Texas. It's going out of control. So think about that on a national level. It's going to be crazy. They won't make the right vaccines. They'll be slow in doing it. They won't be properly studied. You won't know the results. You won't know if they're safe. Oh my gosh. It's horrible. Common chronic diseases like diabetes have pharmaceutical companies that fund R&D. It's the less lucrative diseases that affect lower income countries whose research will suffer. That's true. A lot of the chronic diseases are worked on by pharmaceutical companies because they can treat you for your lifetime and sell a lot of drug. So that's very attractive to them. Kang says, I got the flu in 1968. Am I protected from that version? It depends on your... No, it depends the first flu you ever got. Because if you got an earlier flu, let's say, so 1968 was H3N2. But if your first flu was the previous one, H2N2, then, well, the H2N2 and H3N2 are probably sufficiently different that you made a de novo response to H3N2 in 1968. So you should be protected against that. Elmug1, hello from Pueblos 24C. Hogger51 writes, my tech podcasts have termed what is going on in the USA right now as the enshiftification of America. Oh, enshittification. Yeah, totally. People think they know what they're doing. They think they're competent. They're confident, but they're incompetent. Carol says, enjoyed TWIV 1203. That's, original imprinting and all that. Born in 59, never had the flu. Only a few colds. I find that hard to believe, Carol. Maybe you had some mild influenza. But in 1959, you would be having H2N2. And so that would be your first exposure. And, there's no more H2N2 circulating now, so you're not getting any memory to it. But you're getting, H3N2 and H1N1 are sufficiently different to provoke a de novo response in you. It's only when the strains are similar that you go to the original one that you got. Is a PCR test for H5N1 worthwhile? PCR of what? Birds, dogs, pets, you? Well, if you're not sick, no. But, if you suspect, if you got a bird or a cat that you think might be sick, you could take a swab and do a PCR just out of interest, but you're not going to be able to do anything with that information, right? But, you're a scientist. You could do it. Let's see. El Mugs in Puebla, we saw that. Let's see. This is interesting from Eric. Evolutionary radiation of the bankvole began with divergence of Bardi and Peruzzi clades in their hantaviruses in early 14th century Florence. Both clades bottlenecked when Edward III defaulted on loans for the 100 Years' War. That's very interesting. Why did they bottleneck then? Explain that. That's what I want, a mini lecture on synthesizing and summarizing the TWIV with you, Rich and Brianne. Which question? Yes, a bunch. Let's see. My chat is jumping around. They mean the flu protection and all that. Just tell me, Patricia, what do you want to hear? Bridget's fear is not political. When coverage from insurance reverts to not covering pre-existing conditions, every person that has COVID has had COVID. All other conditions will be deemed pre-X. That's it. Thank you, Silvio Pina, for your contribution to science communication. We really appreciate it, and we need it. Tippy Tops is sending supportive solidarity vibes from a horrified on a daily basis at what you're having to endure from Portugal. A lot of you are relocating here. Wonder why. Yeah, here it's a very nice place. But I'm staying here to fight the good fight. Yep. Hello, Privyet Veronika. +3 and raining in Moscow. SRR, thank you for your contribution to science communication. Love it. John says you were heard and understood in Alaska. Thanks for the microbe TV sticker. Wow. Don't you have to vaccinate your pets? Yeah, against certain viruses for sure. Yep. Alan in Las Cruces, welcome. 2 items regarding immunity. How to identify the 3% of vaccinated persons who develop measles symptoms. If infected, do these have the same immune amnesia? So the people who are vaccinated, in the study of immune amnesia, there was no loss of immune memory in MMR vaccinated kids. So I don't know if that included any that actually had measles symptoms. Maybe that makes a difference. I don't know if there are any studies on that. It's a good question. Arthur asks, do you have any opinions on Gardasil? Gardasil is a fabulous vaccine that prevents cervical cancer, prevents thousands of deaths annually in the U.S. and many other countries. It does not do anything bad to you. It is completely safe, with the usual side effects. Yeah, there are lawsuits spearheaded by RFK Jr. just to make money. And they have an ad. If you were injured by Gardasil, call us. Those billboards on the highway. If you're injured in New Jersey, call us. How would you know that you were injured by Gardasil? Let's see. I had my Gardasil vaccine, and then I got hit by a car and my leg is broken. Really? You got injured by Gardasil? That's the kind of thinking people do. Meritless lawsuits, but they consume time and money. Gardasil is a great, safe vaccine. It prevents cancer. There's no question about it. Everything else is lies. Oh, my God. Yes, no basis to the lawsuits whatsoever. Yet they go on because the lawyers make money either way. Disgusting profession all around, especially for product litigation like this, vaccine litigation. All right. I promised a mini lecture, so let's do that. I will mark the last comment here, and we'll do a mini lecture. I wanted to talk about assembly of viruses, the last part of the infectious cycle, right? How you put them together. So if you look at a virus, you should be able to know how it's formed. So we have a lot of viruses here. Many of them have membranes. So after today's lecture, you should be able to know how they got that membrane. And then there are viruses that are made of only protein without a membrane, like adenovirus and poliovirus, etc. And you'll know how that's formed as well. So you can just look at it and say, oh, yes, here's the pathway. Now, there are always minor differences, but the principles, that's what matters. So all viruses have to go through a set of assembly reactions. So we're at the end of the reproduction cycle, right? We're making, say, protein subunits of a capsid. So these proteins are made. the shell is assembled, and somehow the genome is put into the shell. Those are steps common to all viruses. Some viruses then get an envelope before or concomitant with release from the host cell. The virus particles mature, and then they go on to infect a new cell. So everything is very common except this envelope, which is found in some viruses and not others. So one of the interesting things about assembly is that nothing happens fast in dilute solutions. The protein components that go to make a virus particle are always concentrated somewhere. And here are two examples of that. We call these factories or inclusions. And for poliovirus, the proteins are concentrated on internal membranes. So here on the left is a poliovirus infected cell. And all those little dots are clusters of newly synthesized polioviruses. And these are double membrane vesicles that have been induced by infection. And the particles have been assembled on the surface of them, and the RNA has also been copied on the surface as well. And for rabies virus, which we mentioned briefly tonight, there are very characteristic inclusions called Negri bodies. So here is a neuron that has been in an animal that's been infected with rabies virus and probably killed because this is, taken from the brain. So this cell here, this kind of pyramidal cell, is a neuron. And it's in bad shape. It's got a space around it. So it should be touching all the other cells in the brain. So it's dying. And within it are these circles, which are Negri bodies, which is where the rabies virus is being assembled. Okay? And that was discovered by an Italian pathologist years ago, Negri, Golgi, etc. So he said, these are very interesting. He didn't know what a virus was, but he said, these people who have rabies, they all seem to have this. And now we know it's where the virus particles are assembled. So viruses do their assembly in concentrated areas of the cell. The way many viruses are assembled is by what is called sub-assemblies. Sub-assemblies. It's like an automobile production line. You move along the line and things are added. So you have discrete intermediate structures. So here we're making a bacteriophage. So first we're making the tail. So we make a base plate of a variety of proteins. We make this shaft, and then we cover it with additional discs, and we end up with a tail. Meanwhile, we're making a capsid, which then gets the DNA genome in it, and it's attached to the tail. And finally, we've made tail fibers with a bunch of steps, and those tail fibers are stuck on. So you think of it, the car is moving down the assembly line. Somewhere else, someone has made a door, put all the parts together, and now they stick it on the car. Why would they do this? Why would they have this come out during evolution? I think it's for quality control, because if you make a mistake here in the tail, you can throw it out or reverse it and start over. So it's only at the end that everything comes together. So you have multiple steps for quality control. So that many viruses do this. I think all viruses do this subassembly thing. So here are some ways it works for other viruses for making subassemblies. So sometimes the subassemblies are made from individual protein molecules. So here is the SV40 pentamer. The virus particle is made up of multiple copies of a pentamer, which is 5 copies of VP1, virus protein 1. And the virus VP1 is simply made on ribosomes, and these self-assemble. And so it's a subassembly because now this is going to go on to assemble into a virus particle. Poliovirus is a similar situation, except the ribosomes translate the RNA genome and they make a long protein of the capsid proteins, VP1, 2, 3, and 4. These fold, and then they're cleaved by proteases, and this gives you a structural unit. So that's going to then be assembled into virus particles in subsequent steps. So again, it's a subassembly. And finally, the adenovirus particle is made up of trimers of the hexon proteins. These are made initially as individual proteins, and then they're assembled into a trimer. And this gets assembled into a virus particle in a series of steps. So these are all these viruses do sub assemblies. Here's the whole assembly pathway for poliovirus. So we just finished making a structural unit, right? The 3 capsid proteins together, and 5 of these make a pentamer. And then the pentamer associates with the genomic RNA, and 12 of those pentamers form the virus particle. And then it undergoes a subsequent cleavage to make it infectious. So that's the subassemblies, the 5S, the pentamer, and then the capsid. Same thing for herpes virus, except it happens in the nucleus. So herpes viral DNA reproduces in the nucleus. So the capsid proteins are all brought in to the nucleus to encapsulate the genome. And these are all assembly line subunits, all the components of the capsid, pentamers, hexamers, all these other proteins, a portal, which there's one on each capsid. They're all brought in the nucleus, and they're assembled into what's called a procapsid. And this has a protein scaffold inside it to keep it structurally intact. When it's built, there's a protease built into one of these proteins that digests them all. And now the capsid is empty, and the DNA can be put in through the portal, and all those protein digestion products are gone. So again, it's a sequential series of steps. You've made all these individual subunits, then you build them into a virus particle. Here's an example for influenza virus. It's a little bit complicated because the viral RNAs actually reproduce in the nucleus. Of course, then they have to be copied into mRNAs, which go out into cytoplasm to make proteins, including the HA and the NA, which go to the plasma membrane. And the RNA segments that have been reproduced in the nucleus go to the plasma membrane and stimulate the process of what we call budding. The membrane pinches out and off comes a virus particle. And so the influenza particle is made, again, by these sequential series of steps, and all comes together at the end in budding. Okay, so those are examples of sequential assembly pathways, which are common to most viruses. Another thing that's very important is genome packaging. The problem is that the viral genome, which could be DNA or RNA, has to be distinguished from cellular DNA or RNA when assembly takes place. You don't want to package cellular molecules, right? You want to package viral nucleic acids. So the solution has been packaging signals in the viral genome, whether it's DNA or RNA. And I'll give you an example of a couple of packaging signals. So here's some DNA genome packaging signals. On the top is adenovirus. This is the left end of adenovirus DNA. It's 36,000 bases long. And here are the packaging sequences, these blue stripes. So what do? So in the infected cell, we make first an empty capsid, very much like we did with herpes virus. And the viral DNA is put in. These packaging signals interact with viral proteins, including these that are listed here. And that ensures that they get into the inside of the capsid. So anything that doesn't have these packaging sequences are not going to bind the viral proteins, so they're not going to get in the capsid. Same thing with SV40, which is a circular double-stranded DNA genome. At the origin of replication here, just to the left, there are multiple packaging signals. And again, that makes sure the DNA gets into the capsid. Packaging of herpes DNA is very interesting, so I wanted to tell you about it. If anyone's listening still, I know you're really interested in MMR, but we have to have little diversions like this. It's okay. You can chat amongst yourselves. So when herpes viruses replicate their DNA, they do rolling circle. Remember the toilet paper metaphor. You make long DNAs with multiple genome copies. And at the left end are the packaging signals here, PAC1 and PAC2. So what happens is you have an empty capsid with a portal. And there are proteins that associate with the portal that recognize the packaging signals here. They bind the viral DNA and they start to pump it into the cell under very high pressure. And because this long DNA is repeated, eventually they're going to reach another packaging signal. And when that happens, the DNA is cut. What's left is released. And now you have a unit of viral DNA in cytoplasm. In the virus particle itself. So a very clever use of two packaging signals. By the way, this portal has an active pump that pumps the DNA in, and the final pressure in this particle is 600 pounds per square inch. 600 psi. Try blowing up your bike tires to 600 psi. I don't think you'll get very close. It's amazing that the capsid can handle that. Of course, it's very small, and that makes a difference. But that means when the virus particle sits on the nuclear pore, the DNA shoots out into the nucleus because of that pressure. Very, very cool. Okay, in RNA genomes, we have packaging signals. Here's one for HIV. A little bit complicated, but at the left end of the genome, we have the translating AUG. And there's a packaging sequence called Psi that is around the AUG. It's expanded here. So there's our AUG. That's the initiation of translation. And it's got a bunch of stem loops here. And this one, SL1, has a series of bases that can base pair with another copy of the RNA genome. It's called a kissing loop complex. So here are two RNAs that can base pair via this loop sequence. And so that now is bound to the nucleocapsid protein that recognizes this packaging sequence, and that enables it to get into virus particles. So cells, again, do not have these packaging signals, so only viral RNAs are encapsulated. All right, so I want to show you how this happens. And the main structural protein of some retroviruses is shown here. It's called GAG. It includes the matrix, the capsid, and the nucleocapsid protein. Remember, nucleocapsid is able to bind the RNA via these packaging sequences. Segmented genomes are another story because you have to make sure you get the right number of genomes in. And if there are 8, as for influenza virus, you have to get all of the right 8 of the right ones in. The problem is for flu, a random packaging mechanism, just grab 8 segments at a time from the infected cell. That would give you 1 infectious particle per 400, which is about the known ratio of the virus, 400 to 1. However, there is evidence for specific packaging signals on each RNA segment, and those are shown here. These are the 8 RNAs of influenza virus, and at the ends are packaging signals shown in blue, which interact with viral proteins to get them, these RNAs, into the virus particle. So you always have 8 RNA segments. You always have 1 of each. And the segments are actually oriented parallel to each other, and they bud out of the virus, out of the cell, with these RNAs in an array like that. And finally, there's an interesting example of what we call selective packaging. Where this is a double-stranded RNA virus that has 3 segments. So the segments are called SM and L, and they have a serial dependence of packaging. What does that mean? Well, first, you have to package the small segment. It's the only one that can go in first. It's got signals on it that interact with the viral proteins. And then M will only go in if S is already in there. And then finally, L will only go in if M is already in there. So it makes the virus only package viral RNAs, but also it makes sure that it gets all 3 of the right ones. And this virus has a particle to PFU ratio of about 1, which means that every particle is infectious, as opposed to, influenza virus, where 1 in 400 are infectious. So this is the way to do it, serial packaging apparently, but not every virus does that. This is a bacteriophage, and well, I guess the solution doesn't work for everyone, right? Okay, the last thing we're going to talk about is getting an envelope, a membrane on the particle. So we've briefly mentioned, that viruses acquire envelopes by budding, a process where you acquire the membrane. And so this could be driven by viral proteins. It can be driven by just spike proteins, for example. And we're going to look at how this happens for a bunch of viruses. So influenza viruses, we've already seen bud at the plasma membrane. The glycoproteins are brought to the plasma membrane, HA and NA and others. And then the RNAs, all 8 of them, come to the plasma membrane, and that whole structure buds out to make a virus particle. So this one buds from the plasma membrane. So how does the RNA know to go to the plasma membrane? It's pretty far away, right? Well, first of all, it's brought there on microtubules by motors, okay? It just doesn't dissolve, diffuse. But the M1 protein has hydrophobic regions, and those are what are attached to the RNA, and those M1 proteins bring the RNA to the membrane, because hydrophobic regions of proteins like to be next to greasy membranes. Same thing for VSV, another RNA-containing virus. There are hydrophobic regions that bring the RNA protein complex to the plasma membrane. Now, retroviruses are very interesting because they mature after they're released from the cell. So remember, we have a dimer of the RNA genome, which is brought together by those kissing loop sequences. And these RNAs bind the viral protein. And remember, the protein is made GAG protein is made as a precursor, matrix capsid, nucleocapsid, and the nucleocapsid part of this precursor binds the RNA, and then the matrix has a hydrophobic region attached to it as well, and that brings it to the plasma membrane. And all of these molecules line up. So a couple of them bring in the viral RNA, but the rest of them just line up to make a capsid and a M protein. And sometimes the polyprotein gets a little bigger, and it includes reverse transcriptase and integrase, which you can see here is being brought in. So all this assembly occurs at the plasma membrane, and it buds off to make a virus particle. But only after it buds off, and you can see you've got some reverse transcriptases in there, but mostly not, and 2 molecules of RNA. Then there's a protease in here as well that cleaves these proteins to make the icosahedral capsid. That's the final maturation step, so it actually occurs outside the cell. Very cool. So again, the matrix protein of retroviruses is actually myristylated with a lipid at its end terminus, and that targets it to the plasma membrane. And finally, we have to mention SARS-CoV-2, right? Because not all viruses bud from the plasma membrane. SARS-CoV-2 buds into the ergic, the ER-Golgi intermediate compartment. So the genome is replicated in the infected cell. It's attached to the nucleocapsid protein, and that buds into the ergic, where the spike and other membrane proteins have been inserted. Now you have a vesicle with a virus particle in that's goes to the plasma membrane and fuses and releases the virus particles. So the budding is into the ergic, not from the plasma membrane. Now, viruses can bud from any membrane in the cell. So plasma membrane, ergic. Here you'll see multiple membranes for herpes, but it's always consistent within the virus. It doesn't change from one day to another. Herpes virus capsids are assembled in the nucleus. So there we have a capsid with DNA. They bud out of the nucleus through the nuclear membrane, and they acquire a membrane. Now they're in the endoplasmic reticulum. They bind to the ER membrane and fuse, and now we have a nuclear capsid in the cytoplasm without a membrane. Not good. So this structure buds into the trans-Golgi network. It acquires a membrane there. It eventually leaves the trans-Golgi in a vesicle and then fuses at the plasma membrane. So if you look at herpes, the final membrane around the particle comes from the trans-Golgi network. So lots of different ways that viruses assemble, but we've learned to study them independently and get consensus on how things happen. Okay, that's our mini lecture for tonight. Oh, and we still have Tom up here. So let's go back to the chat. Let's see, I talked about Gardasil. So Eric elaborates on the vole story. In the UK, Bank of England vole and associated thread needle street virus restricted to a small area of the City of London. TSV spread by contaminated banknotes as hypothesized to have caused an outbreak in the 70s. Very interesting. Margot says it seems like people aren't fighting out of fear or of being targeted or of losing their job. So many science jobs are vanishing. Yes. People have to start fighting, right? Well, the professors are going to be okay because they're tenured and they're paid by the university. But the people who work for them in the labs are gone because their grants are gone. Martha says, how do we know that a measles infection will provide lifelong immunity? Aha! The Faroe Islands. It's not because you have been tested. It's because in the 17 and 1800s, a small island community in the north of the UK was tested. So I think we should bring up, where is the Faroe Islands? This is going to be an immune memory. Okay, so we'll look up that, adaptive immunity, that is, 2025 lectures. Adaptive immunity would be lecture 14, I believe. It's a good question, Martha. There we go. Memory. I will get to the Faroe Island slide. This is very nicely described in Adam Ratner's book, Boosters. But I'll do a brief description for you. So, in 1781, there was an outbreak of measles on the Faroe Islands here between Iceland and Scotland. People only went there by boats and not very often. And so this outbreak, and remember, they didn't know this was a virus. They just knew it was some kind of contagion. This outbreak was presumably brought in by someone on a boat, brought from the mainland. For the next 65 years, there was no measles on the island, right? Why? Many people were immune. So that's lifelong immunity. In 1846, there was another outbreak. None of the people who survived the 1781 outbreak contracted measles. So from 1781 to 1846, that's how long immune memory lasts. Now, okay, so that's the experiment that initially showed this. I'm going to show you this graph, which is really cool. The persistence of antibody for different viruses. Persistence of antibody, like in your blood, indicates the presence of memory B cells. So look at this. These viruses that cause systemic infections, they spread in your blood. 30 years, 75 years for yellow fever, 65 for measles. That's from the Faroe Islands. 75 for vaccinia. Amazing. Polio, 40. But then you go to mucosal infections. 12 months, 30 months, 3 months, 12 months. Why is that? We don't know. But some viruses produce longer lived memory than others. Anyway, I hope that answers your question, Martha. Yes, the orange guy. That's what we call him here. Will says, well, I'm disgusted by the orange regime. I do want to reiterate that I spent my whole life living and working with the most wonderful Americans. Thank you, Will. There are many wonderful Americans in this country, but at the moment we're outnumbered by people who I would say are not wonderful. They're more interested in themselves. Yes, Marco says the Call 5 app is excellent for contacting your representatives to voice your opinions about the unreasonable changes which are being attempted. This is interesting. Mark read a number of years ago that non-smoking lung cancer was linked to a null mutation to TLR9 and infection with HPV. I have never heard that. But according to the literature, HPV 16 and 18 can be a contributing factor to lung cancer. Can cause chronic inflammation in the tract, leading to DNA damage and the development of precancerous lesions. So this is not the same mechanism that would come into play in mucosal epithelial cells where you have an oncogene in the viral genome. And so, but yes, vaccination would prevent that, yes. Although I don't know that the vaccines have been tested, so they would have to be used off-label. But you could get HPV vaccines if you're up to a certain age in men or women, and that would protect you against that. Interesting point. We've solved problems for so long that people have forgotten why we need to do anything about them. So that includes the vaccines, right? But, all the efficient running of this, and this country was run very efficiently up to recently. The Social Security Administration was not overstaffed. It hasn't increased its personnel in 50 years, yet he decided to cut X percent of the workers for no reason. It's just ignorance. But, yeah, people forget that their Social Security office runs really well. Now go in and try and get a claim. Forget it. Forget it. Truth or Dare writes the article in the Irish Times with off it states MMR infant vaccination rate is down to 85%. Other countries are low, with Japan at 54%. So that's a problem. We're going to, there's a lot of measles globally already because many countries don't vaccinate enough, but it's going to get up. It's going to go higher and higher. Yes, we need a new Richard Feynman. Absolutely. Mary Nika says, watching American scientists and other institutions gives me flashbacks with how it started in Russia. Please fight back while you still can. Don't just hope that it'll be over in 4 years. Oh, I agree totally. It's not going to be over in 4 years unless we do something, because they're going to eliminate the ability to vote, and we're not going to be able to get rid of them. They're going to be with us forever. So we do have to fight. People need to realize this. And these upcoming special elections are really important. But I'm just not so confident that they're going to do what we need them to do. Ferrets are not people. That's right. Mice lie, monkeys exaggerate, and ferrets are not people. Tractor Poodle says, I hope I'm wrong, but we could be facing civil unrest on a level that makes the 60s look like kindergarten pillow fight. Yeah. Yeah, so I don't know, Tractor. I think many people have gotten complacent, as Bridget points out. So I don't know. But the response is going to be horrid. The police will show no restraint. Horrible. Vista asks people who get it H5N1 conjunctivitis, could they get immunity to the MAGA H5 strain that spreads human to human? Yeah, they should. As long as it's similar enough, they should get an immunity to that infection. They're infected. They're making antibodies and T cells, yeah. Veronica suggested discussion with Dr. Griffin. Some info about that medication, which was supposed to be better than Paxlovid, but its funding has just been taken away. I'll mention that to him. Yes. Angry Penguin, worst vaccine I ever had was rabies. So in the old days, you used to get 12 rabies shots in your belly, your abdomen. In the, not intraperitoneally, just in the muscle. But I never got that. But I heard that was the most painful thing, because after the first few, it was all inflamed, and it would hurt so much to get more and more and more. 12! Oh my God, I'm so glad they changed that, right? Wonder if H5 would survive in milk. It does. They've done experiments in mice. They take cow's milk from cows who are infected, and they feed it to mice, and the mice get infected. So it does survive. Yeah. It's a concern. And, they sell milk that's PCR positive. All right. Once the cows recover, they could still be shedding virus, and they can sell the milk. And you would be surprised how much PCR positive milk there is out there. Martha thinks that Congress is bribed. I don't know of any evidence for that. I just think it's power. It's a power thing. How about a mini lecture on those mutations? You're talking about the H5N1. Yeah, I did one not too long ago, but we could do another one. Sure. I'll do anything. I like talking about viruses. Yeah, this is a good one. A new definition for outliers. Very good, Nevitz. Oh, here we go. The Gibson novel, The Peripheral. This is why we can't have nice things when she witnesses a crime. It's a very characteristic phrase. Yeah. Any thoughts on Side Tech Daily's article, Universal Coronavirus Vaccine Breakthrough, a single shot that could protect you from COVID, MERS, and the common cold? I have to look at this, Jeff, because common cold is a bit much to be part of that. And I don't know who's developing this, but if it's in the U.S. and it's supported by NIH funds, I don't think it's going to get anywhere. But let me take a look at it. If you could paste, well, you can't paste the link in here. I'll find it. Oligarchs may have hospitals if they get sick, but where will the scientists be to cure them? Well, the scientists will be paid very handsomely, and they won't have a choice but to take care of the oligarchs. Yeah. Wonder how much mercury is in cod liver oil. You can probably figure that out. Yeah. If there are asymptomatic H5 in humans, that would skew the mortality rate. Yes, and that was a big argument years ago. Peter Palese used to say there are a lot of asymptomatic infections. This 50% is nonsense. And because of the places where there are a lot of chickens next to people, the healthcare is crap. So you wouldn't know if they had asymptomatic infection. So I think that's a really good point. Yeah. How do we get the politics out of science? You can't, because science is funded by the government, which is politics. And so, if you have a reasonable Congress, it's fine. But now we have an unreasonable one, and it's done. Okay, let's wrap this up here. Lots of great questions here, by the way. Really appreciate it. Yeah, measles used to be under control in the U.S. Now it's not. John, thank you for your support of science communication. Made progress with someone fearful of childhood vaccines in the FDA using info I learned here. Not as much on pharma because of a checkered past there, but I'll take any small wins these days. Thank you, John, for doing that. Really appreciate it. Do you support the idea of presenting both good and bad papers during journal clubs? I do, so that you can see what went wrong. I mean, not woefully or not bad on purpose, but just errors of interpretation. I think it's a good learning experience to do that. Yeah. Well, the discoverer of HPV, what was his name? I interviewed him. Yeah. Harold Zurhausen. I interviewed him. He got the Nobel Prize, but no vaccine developer has got a Nobel Prize. Yeah. Okay, a lot of good questions about assembly. Do most antiviral drugs mainly prevent viruses from assembly? No, actually they don't, but there is a Hep B drug that prevents assembly. It's very unique and it's very effective. That might be coming up in an interview from the HPV meeting that we're releasing soon. Yep. Yeah, how do they measure the PSI in a virus? They do. I don't remember offhand how it is, but I'm thinking of very, something like atomic force microscopy. They could use that. Yep. Hey, MN says the same tiny velvet bag where you tuck in the tiny tire gauge that measures tiny but very high PSI. That's just lovely, MN. Really, really good. I love it. Thank you, Questella, for your contribution to science communication. Really appreciate it. This is true. Research is prosperity. Seek to create an intellectual trade surplus by luring the brilliant with funding, and you will grow industry. The return on investment of NIH is 3 to 1. It's always been great, and anyone that tells you otherwise is wrong. And yeah, industry as well is really important. Heather, thank you for your contribution to science communication. I really appreciate it. Help us keep going here. All right, that'll do it for another office hours. I want to thank our moderators, Steph and Les, and who else was here? Barb UK was here. Tom Steinberg was here as well. Thank you all for joining us tonight and keeping it civil. And thank everybody for coming. We have 200 people and 200 likes. Love it. Love it. When are we going to be back? Next week, actually. We need to do this every week because we need to keep everybody engaged in the science. So today is the 26th. Next week, it will be, oh my gosh, April already. April 2nd, 8 p.m. Eastern Time. Okay, folks, thanks for coming. You really enrich the office hours. I love your questions. I love your bantering amongst each other. And come back next week, please. In the meantime, be safe. Good night.